Hello students, I am Dr. Vinod Shinde. We have discussed in the previous lectures about the HMP, EDP, KP and EMP pathway along with pentose phosphatidylose pathway, hexose phosphatidylose pathway, TCA cycle and respiratory electron transport system, in which we have also discussed the interrelation between these pathways. Now we will discuss about the fermentation and type of respiration. In the fermentation cycles, respiration is an ATP generation process in which either inorganic or organic compounds function as electron donors and inorganic compound serve as a ultimate acceptor. The term aerobic respiration, anaerobic respiration and fermentation are often in discussion on energy yielding metabolism. In aerobic respiration takes place in the presence of free oxygen, which is the final electron acceptor. Most organisms are strictly aerobic. They are totally dependent on the oxidative phosphorylation and cannot survive in the absence of oxygen. In the aerobic respiration, however, energy is obtained from oxidation, in which inorganic compound function as final electron acceptors. The ultimate electron acceptor are compounds like sulfates, nitrates, carbonates. Although these compounds contain oxygen, it is not present in the free form. Some microorganisms utilize oxidative phosphorylation when possible, but can also survive in the absence of oxygen, that is called anaerobic respiration. They generate ATP by anaerobic respiration with the nitrates or by fermentation. Such organisms are called facultative anaerobes. They burn fuel when oxygen is present, but can resort to anaerobic respiration in the absence of oxygen. In the strict sense of fermentation refers to those energy yielding pathways in which organic compounds act as both electron acceptors and donors. When the compounds donate electron, they become oxidized, and when they accept electron, they become reduced. During fermentation, microorganisms obtain energy from organic compounds without utilizing oxygen. The compounds which serve as electron donors acceptors are usually two different metabolites derived from single substrate such as sugar. The process of fermentation takes place in two stages, that is, glucose is broken into pyruvate with the release of two pairs of hydrogen atoms. Pyruvate is or compounds derived from pyruvate are reduced by the released hydrogen in the first stage. Now we will discuss about the alcoholic fermentation. In the alcohol fermentation, pyruvate is converted into ethanol and carbon dioxide. In this process, the characteristic of yeast and particularly strains of Saccharomyces cerevisiae, it is also found that some molds are also used, and it is comparatively rare in bacteria. In the bacterium, Pseudomonas is used. Pyruvate is produced through the ED pathway. It is then metabolized to ethanol through acetaldehyde. A molecule of glucose yields two molecules of pyruvate through EMP pathway. Pyruvate metabolism takes place in two stages, that is, first stage of decarboxylation, where pyruvate is first decarboxylated yielding acetaldehyde and carbon dioxide. The reaction is catalyzed by the enzyme pyruvate decarboxylase with thiamine phosphates (TPP) as a coenzyme. Acetaldehyde is then reduced to ethanol. Acetaldehyde is reduced to ethanol by NAD/NADH2, and NAD is regenerated. The catalyzing enzyme is alcohol dehydrogenase. It will be seen that hydrogen removes during the glucose metabolism and accepted by NAD, which is reduced to NADH2. It is essential that NAD is regenerated so that it will pick up the more hydrogen, and if it does not happen, glycolysis would stop, resulting in the death of the organism. Regeneration of NAD takes place when acetaldehyde is reduced to alcohol. Alcoholic fermentation is the economic importance in the production of beverages and raising breads. Here if you observe, glucose is converted into pyruvate through EMP pathway, where NAD is reduced to NADH2, that is 2 hydrogen atoms. Now this reduced NADH2 is regenerated to NAD, while acetaldehyde is getting converted into ethanol. The pyruvate is converted into acetaldehyde with the release of carbon dioxide, and acetaldehyde is converted into ethanol in the presence of alcohol dehydrogenases, where the reduced NADH2 is again regenerated to NAD+, which is important for the alcohol fermentation and glycolysis to continue. In the alcohol fermentation, it is of economic importance and is the major product produced in the industry. It is consumed as a beverage or also used for the fuel, and is good. This fermentation process is good in the food industry as well as in the dairy industry. So this is the alcoholic fermentation. In the next lecture we will discuss about the lactic acid fermentation. Thank you.